Hi, my name is Divyan Seni and I am working on this project with my partner Lanea and Mansimrat. And our aim for this project is to check the impact of gibberellic acid on stem length in normal and dwarf pea plants. And the species that we worked on is Pisum sativum. So the introduction of our research project was to aim to investigate the influence of gibberellic acid, aka GA, which is a hormone found in plants for on the growth and dwarf pea plants. So the two with the same species and two varieties was Pisum sativum, and the two varieties we worked on was Little Marvel and Ederson, which is normal pea plant. The experiment is motivated by understanding that gibberellic acid plays a crucial role in plant growth and development. The hypothesis for our project was if the different concentration of gibberellic acid are applied to normal and dwarf pea plants, there will be a corresponding increase in the length of both normal and dwarf pea plant. It's definitely because of the growth promoting properties associated with gibberellic acid, which expect to enhance the elongation of the pea plant. So it just basically means that our hypothesis is suggesting that if you apply different concentration of gibberellic acid to both normal and dwarf pea plants, it will give result in increased stem length due to the GA known's growth promoting properties. The experiment for our independent variable, the manipulating independent variable, which is the concentration of GA, we use two different concentration. The first one was low gibberellic acid, which is 0.5 gram, and high gibberellic acid, which is 1.0 gram. The dependent variable for this was stem length and internode length for both normal and dwarf pea plants. There were other control variables also implemented to ensure the validity and reliability of the results, and we also did reducing bias in the experiment.
As discussed previously, gibberellic acid plays a really important role in plant growth and development processes. The background on this is that Japanese scientists in the 19th century discovered that rice plants infected with Gibberellia fujikuroi, a type of fungus, grew significantly taller than usual. Gibberellic acid's effect on stem length in both regular, so normal pea plants, and miniature pea plants, which is the dwarf, must be understood in order to understand why genes and hormones interact to drive plant growth. Research has shown that GA promotes cell elongation and division, which leads to increased plant height.
Gregor Mendel, an Austrian botanist who studied in the field of genetics and biotechnology, has performed several experiments related to pea plant growth and stem elongation. And one of his significant studies that he performed was related to stem length, for which he found out that the stem length is regulated by just a single gene. He discovered this by examining how tall the plants grew. He experimented that on different varieties like the dwarf, which one was the Little Marvel, and the normal variety, which we call Anderson. The dwarf pea varieties often grow to a height of less than 1 meter. They are essentially shorter versions of the normal or tall varieties.
Then we did our methods for the project. So we did the experiment setup, which involved 18 pods, which we divided into 6 groups. 3 pods per group to study the GA on stem length. So we did 2 varieties, which we discussed already in the introduction. After that, the pods were filled with uniform soil, and we assigned specific treatments. There were 3 kinds of seeds which were involved, water soaked, low GA concentration, and high GA concentration soaked seed for each variety. Then the plants were placed by the window, and we monitored them as it was necessary. The stem length were measured when the stems were developed. The data was also recorded with clear labels for experiment groups, GA concentration, and plant types. The statistical analysis we used mean by taking the average, and we also did standard deviation by making a bar graph, which was conducted to interpret the result and compare the effects of GA and plant types on our stem length. We also did bias reduction steps, which include controlling variables taking multiple measurements for our data and ensuring consistent plant throughout the experiment.
So during the analysis of the impact of gibberellic acid on our pea plant growth, we had a challenge with poor pea germination, which resulted in minimal growth with our data. By the end of week 3, only a few plants had notable growth, which was the 2 marvel plants treated with low concentration of GA, which reached heights of 5.1 centimeters and 1.9 centimeters. And an Anderson plant that was treated with high concentration of GA grew to 6.1 centimeters. And the little marvel plants without GA showed the most growth, reaching heights of 11.2 centimeters and 2.1 centimeters. And similar heights were observed in week 4. So we had to have supplementary data provided to support us and further analyze our experiment outcomes.
In the supplementary data provided, we saw that the plant growth over 3 weeks revealed distinct patterns based on different concentrations of gibberellic acid. The little marvel plants treated with the high concentration of the hormone demonstrated most significant growths. They reached to average heights of 46 cm by week 3. On the other hand, Anderson plants without the hormone exhibited the lowest average heights across the experiment, measuring only 24.1 cm. Little marvel plants treated with low gibberellic acid concentrations showed an initial growth of 2.2 cm in week 1, which declined to 20.1 cm in week 2, and it just reached to 38.1 cm by week 3. While in the absence of gibberellic acid, little marvel plants displayed minimal growth with an average increase from 2.1 cm in week 1 to just 16.4 cm in week 3. The Anderson group's growth varied with gibberellic acid concentrations, achieving an average height of 45.2 cm with high gibberellic acid and 38.7 cm with low gibberellic acid by week 3.
So by statistical analysis, we did standard deviation by making a bar graph and showing a result. So our experiment basically highlights the significant difference in our stem lengths in control groups and those treated with varying doses of gibberellic acid. As showed in the bar graph, the treatment with G resulted in increased stem length in both normal and dwarf pea plants comparing to the control groups. By week 3, if you see, the little marble plantains treated with high concentration of gibberellic acid showed a stem length of 46.0 centimeter, which was definitely significantly higher than the controls group average of 16.4 centimeter for untreated little marble and 42.1 centimeter for untreated Anderson plants. Similarly, in the high gibberellic acid treatment group of little marble, the average stem length reached 45.2 centimeters by week 3, which definitely exceeding the controls group average of 24.1 centimeter. And these were the highlights of gibberellic acid growth promoting effects with little marble high of GA group, surpassing the Anderson control group in the stem length due to GA's influence.
The experimental results support the hypothesis that GA's growth-promoting properties would lead to increased stem length in both normal and dwarf pea plants. This table here demonstrates GA's role in promoting cell elongation and division. The data from our experiment is supported by existing studies highlighting the efficiency of GA in stimulating plant growth.
The effects of gibberellic acid on stem length are closely linked to its role in stimulating cell elongation and division in plants. The gibberellic acid hormone promotes the synthesis of proteins that facilitate cell expansion, which result in increased internode elongation and overall growth of the plant. Despite the significant findings, several potential sources of errors were recorded in the experiment, such as poor pea germination, which posed a significant challenge for us to impact, and it impacted the reliability of the data collected, for which we were provided a supplementary data. We saw that each plant received the same amount of water at the same time, but it was noted that the timing and the volume of liquid provided to the plant was really inconsistent.
For example, in the first week, the plant received 17 ml of liquid, which resulted in minimal growth rates. That means inconsistency may stem from insufficient research into optimal seed watering and planting practices. But we did like some adjustments, like providing 60 ml of water to the plants in the second week, which definitely gave us more consistent moisture level and improved growth rate by the third week, as indicated in the table.
To address these errors in future experiments, we decided that careful implementation of controls will be essential. Addressing challenges related to pea germination is critical for ensuring experiment reliability. Variations in environmental factors like temperature, humidity, and light intensity can influence plant growth and introduce variability in experimental results.
To conclude, we would say that gibberellic acid played an important role on pea plant growth. The plants which were treated with the hormone showed increased stem length in comparison to our controls. Identified challenges were poor pea germination and inconsistent watering, which highlights the need for important and careful experimental design. The future research should address these challenges for more accurate results.
And these were the references that we used for our research project, and thank you so much.